we've just seen before that snails that amplify the schistosoma parasites are associated with certain type of floating vegetation in our transmission sites. So with the drone, we started to map our transmission sites, as we show here, through a series of flight plans, a series of overlapping drone photos taken in nadir at each water access point, 32 of them, at 50, 80 meters above the ground level on average. These images were then stitched together using open source software, Open Drone Map, creating extensive georeferenced map of the transmission sites. Our combination of this meticulous on-the-ground snail sampling with concurrent drone mapping of the study sites allowed us to accurately estimate snail abundance and density for a given water access location once we know the extent and composition of the plant species present. And in fact, in a paper that we published in 2019, we show that there is a strong statistical association between the area of floating vegetation per site and the number of snail clusters that are present in that site. We also noticed that snails were extremely patchy and ephemeral and changing in space and time all the time. But it was much easier, in fact, to track suitable habitat than sampling snails in the field. And so by looking just at the area of floating vegetation, it was possible to provide a good explanation of village to village variation in the level of transmission and reinfection. In a paper that previous graduate students in my lab, Dr. Isabel Jones, just published a few months ago on PLOS NTD, we also understood much better how the spatial distance and the geometry might play increase in the risk of schistosomiasis transmission. So we basically now have a better understanding of how far we needed to track the vegetations where the snails that can potentially infect me might be present. So when you look at here, we know now at the village level, basically what we can do, and we know that pretty well. But when we zoom out to the entire lower basin of the Senegal River, the areas is humongous. We are talking about 10,000 square kilometers or more. And it is really important to understand on this heterogeneous landscape where the hotspot of transmission are. So at this scale, there is no drone imagery or drone, if you want, not even those with the fixed wing that can work. So we decided to look also at satellite imagery. Well, the quality is not so good as the drone imagery, but it's improved dramatically in the last few years. And so we can use imagery like the World War II or the Planet Lab to identify snail habitat from satellite imagery. There is no way on the other end we can parse such a huge area manually, but what we can see with our eyes now, we can train computer to do that for us by using machine learning and a specific technique of machine learning that is deep learning. The drone mapping has been still crucial for creating high quality image data. And although we don't have the data for the entire scale at which we want to work, we use the drone imagery as our basically validation of whether the habitat that we were looking at the satellite imagery was indeed a habitat constituted by floating vegetation or emerging vegetation. And so we use the drone data basically to segment the satellite imagery and create a training data set. And then Zach Leo, our machine learning guru, along with Andy Chamberlain and Kurt Italian, used a convolutional neural network and a specific type of pre-training network unit that has been widely used in biomedical application. And we've been able to use actually a very limited number of imagery as training data sets with excellent results and remarkably high accuracy. Precisely, we annotated only 50 sub-images or worldview 2 satellite imagery, 80 by 80 centimeter, to create a binary mask for each class. And we use 40 images, 256 by 256 pixels each, with 8 bands of digital globe, and 10 for training and 10 images for validation. And we use 4 masks for annotation, land, water, emergent vegetation, and floating vegetation. And as we mentioned, we use 80% for training, the 20% for validation. We had a big prevalence of pixels that were labeled as water or land, and 5% as aerial floating vegetation, 10% as emergent vegetation. I skip some of the technicalities just to show you that the accuracy is actually remarkably good. We wanted to test our deep learning approach with more classic machine learning, in this case, like random forest. And what we can see, and I just want to, in terms of accuracy, to go directly to the point, is that on the training data sets, the random forest actually perform in as well as the convolutional neural network that we train. But when we look at the whole doubt validation data sets, the out-of-bag data sets, the convolutional neural network is actually working much better. And so we are finally able to put together a system that integrates what we call the old-fashioned malacology, the one where you step into the water for hard work for hours per day for 3 weeks, and then you go in the lab and check the snails and dissect them and look at the parasites, with high definition drone imagery between 0.5 and 2 centimeter and satellite imagery. And we're now moving into using Planet Lab that is available even on a daily basis for basically the entire globe. And we can integrate this to identify the hotspot of transmission that then we can use to calibrate network models for schistosomiasis dynamics and optimal control. Here we partner with my former mentor Marino Gatto and two of our students back then now professors Professor Lorenzo Mari and Professor Renato Casagrande in Politecnico di Milano, who developed these network models of schistosomiasis. So we had a model for all Senegal and villages. And Lorenzo and Renato use a stratified type of models. And this is really important because this is a big difference with respect to other type of viral or bacterial disease. For instance, here, the number of parasites per host make a huge difference in terms of pathology. And so if individuals has only very few parasites, the damage is relatively limited, but with a large parasitic load, with more than 50 worms per individual host, the damage could be really consistent and might cause cognitive impairment, slowing down on understanding and concentration at school, and gross stunting and bleeding, urogenital infections, up to the development of cancers for kidney, livers, and bladder. So tracking the abundance of parasites per host is actually very crucial. In addition to the human parasitology side, we had a more classic compartmental model for the snail, where we had susceptible snail and infected snail, like we have seen the SI type of model. And then we had also a question to track the shedding of cercaria, the infective stages for humans, so the shedding from the snail into the water, the free living stage, and the other free living stage that is when the eggs that are released with feces or urine by infected people get in touch with the water, they hatches, and they release the metastadia that eventually swim and infected snails of the specific species. In addition to that, Lorenzo and Renato use mobile phone data from Sonatel and Orange that provided in a challenge this information to calibrate the people movement, because people connect at different sites by moving from villages to village or from village to city and so on and so forth. And what they found is that moderate mobility may reduce disease prevalence, but crowding during national events like the religious feast might potentially increase transmission in certain conditions. How much these results depend upon the specific network topology and connectivity of Senegal with respect of, say, the one that we found in Burkina Faso or in Côte d'Ivoire, where we also work. And to understand these questions, and so to take basically a step back and zoom out and think about what are the generalities. I mean, do we have do we need to have phone data, for instance, for each country where we want to optimize the delivery of control treatment, or we can look at the network and understand what's going on. So here, a couple of years ago, we recruited two former undergraduate students, Emily Alstensger and Jonathan Camp, who started to work on look how network topology might affect the transmission of schistosomiasis. And even with super simple network like 3 nodes, with different R0 in each node isolated and different level of mobility, we saw that the location of node with low or high R0 is actually relevant. In fact, if we compute the generalized reproduction number G0 for the entire network, it's the equivalent of R0, but it's for the network, and we plot basically the G0 as a function of the fraction of people moving, we see that depending upon the location of the node, this relationship might be, everything else being equal, might be really different. So understanding topology is probably very important. So we kept building on this, and thanks to Richard De Leo, who is a graduate student who's about to defend his thesis. Now we look at a network of different complexity. We showed before the 3 nodes, but also we look at 15 nodes, 100 nodes, or 1,000 nodes, which is the network, for instance, that Javier Perez and Andrea Rinaldo developed to look at schistosomiasis transmission in Burkina Faso. And similarly to the model used by Perez et al. 2015, which we are as an author, we look at mobility, so again, the fraction of people moving, and we use a radiation model to understand what is the potential flow of people moving over, through different villages and towns in our network. And we use also additional hypotheses on how the population size in a given village or town is an indicator, it's a proxy indicator of the type of association with water, for instance, how much time people spend in natural habitat where we can find the Bulino snails or the Biomphilardo snails that are responsible of transmission of schistosoma, hematobium, and Mansonae, respectively. And so we can scale basically transmission rate as a function of population size of the village. And what Richard found is that in a rural network of villages, people movement increased disease persistence. And so that type of movement, village to village, is actually really relevant to maintain the disease. But when we scale out at the national level and we look at urban network with some very absorbing nodes like Dakar, like the capital of Senegal, well, in that specific case, mobility might actually decrease the transmission because people spend more time in cities and less time with the feet in the water where they can get potentially infected. These are all theoretical considerations that have some importance. The point is, again, what do we do with that? What are the implications for control, especially in rural network where the prevalence of infection is the highest? And so what we look here is that, what can we do if we have to administer protequantal, the drug of choice for removing the parasites from people, in what we call program of mass drug administration. You treat everybody or a lot of people in the population. And we basically look when the case, we say constant means we treat all the nodes at the same level without looking what actually happened in the nodes. So basically treat everything uniformly without knowing any better. Or we can have a backbone, the envelope estimation of what are the prevalence of infection in a village, and treat more the villages where prevalence of infection is higher. We can use other features of the network, like for instance, between centrality, and to see whether a node is really central in the connectivity of our network, whether it's relevant or not. Not surprisingly, if we are using MDA, only mass drug administration with protequantal, yes, we should focus on the villages that have highest level of prevalence. which is actually very important. But if we want to do snail control, our preliminary analysis show that actually the nodes that are mostly connected are on a rural network are probably the one that should be treated more in terms of using fludarabine molluscicide. And of course, when we combine the strategies, if we continue to target nodes with high prevalence with praziquantel and nodes that have a high bitumen centrality with molluscicide, this is where we get the best outcome of all. So, in conclusion to the second part, I want to stress the precision mapping of suitable habitat for the obligate intermediate hot snail might help to identify transmission hotspot for schistosomiasis. This information can be used to parameterize network models. An analysis of network topology and of disease dynamics on a network might help us to identify optimal control strategy for schistosomiasis. And with this, we ended the second part of the seminar, and we're now moving on to the third part.